In this video, I'll give you some tips for Lab 8 enzyme activity. In this experiment, we are working with the lactase enzyme, which works on the lactose disaccharide. Lactose is a disaccharide made up of two glucose molecules. So in the presence of that lactase enzyme, the lactose disaccharide undergoes hydrolysis. It splits apart to form two separate glucose monosaccharides. You are going to be testing the extent to which this reaction occurs by monitoring the amount of glucose that is produced in a bunch of different solutions. And you'll be monitoring the glucose production using little test strips that measure the concentration of glucose. So in part 2, in the first experiment testing that you're going to do, you will have 3 different disaccharides: lactose, maltose, and sucrose. And you are going to put the lactase enzyme in each one of those solutions and then test them to measure how much glucose is present in each one. Now, of course, we expect this one to have a relatively high concentration of glucose because we know that the lactase enzyme works to help perform this experiment. The lactose molecule with the enzyme present is going to be split apart. So we should expect to see a pretty high number of glucose in this box. And our question is, does this lactase enzyme also work on other types of disaccharides like maltose and sucrose? Will it also produce high concentrations of glucose? So in these boxes, you'll just be reporting the numbers that you get from the test strips. And if you want, you can jump straight ahead to the post lab. Question number 1 is asking, is lactase a selective enzyme? That means by selective enzyme, we mean, does it only work on lactose, or does it catalyze the hydrolysis of the other disaccharides that we use? Now I'm trying to remember maltose, and I don't remember what the third one is. But you're going to be answering this question based on the numbers that you got from the test strip. Was the amount of glucose produced by maltose and whatever the other guy was there a lot of glucose for those? Yes or no? If it produced a lot of glucose, then that means that, yeah, it's doing a pretty good job as an enzyme for other types of monosaccharides. Now for part 3, we are going to be testing how well lactase works under a variety of pHs. You have 4 different test tubes that are approximately pH 2, 7, 8, and 12, although you will be reporting the actual pH values of these solutions. And then in this one, you're only going to be working with the lactose solution. You're not going to be working with any other type of disaccharide. You will be putting the enzyme, the lactase enzyme, into your lactose solution, and then using your test strips again to measure the glucose concentration in each test tube. Our question here is with the lactose, again, turning into 2 glucose monosaccharides. Does this reaction work equally well at all 4 of these pHs? If it does, we would see pretty much the same amount of glucose at every pH. Or does this reaction work particularly well at just 1 or 2 of these pHs? So let's just say here that our pH 12 was the highest amount of glucose, and that would tell us that at this particular pH, the enzyme works really, really well because we've produced a whole lot of glucose at that particular pH. With that information, you can head on over to post-lab question number 2 and answer this. Is the activity of lactase sensitive to pH? So being sensitive to pH would mean that it would have you would see different glucose concentration at different pHs, different pH values. If so, which pH was the most active? That would be the one that produced the most glucose. And which pH value was the least active? That would be the one that had the least glucose. Then we're going to take a look at how temperature affects lactase. You will be performing this experiment under 3 different temperatures: ice water, room temperature water, warm, and boiling water. Same kind of thing. You'll be working with just the lactose solution. You'll put the lactase enzyme in there. You'll use your little test strips to figure out how much glucose is present in each one of these different solutions. The concentration of the glucose is going to let you know, like let's say that this is the highest. The solution that has the highest glucose concentration is the one where the enzyme is working the best. Remember that this enzyme is causing this reaction to take place, and so the more glucose we have, that means the better this reaction is working. So if this was the data that you got, if this was the highest number, that would tell you that under ice cold conditions, this reaction is working really well. The enzyme is working really well. Head on over to the post lab. This question number 3, pretty similar to what we did for question number 2. What is the relationship between the enzyme's activity and temperature? And you would answer this something like the warmer it got, the more effective the enzyme was, or the colder it got, the more effective it was, or whatever kind of observation you made. And then, last but not least, we're going to be looking at the effect of a few different substances that have the potential to be an inhibitor of the lactase enzyme. You'll be, again, working with just the lactose solution. You have 4 different test tubes. One of them is the control, and this we would expect to have the highest glucose concentration. And then we've got these 3 substances that all have the potential to be inhibitors. Now, if they are inhibitors, so let's say galactose is an inhibitor, what we would see here is some number that is lower than our control. Remember, again, the reaction that we're looking at is the lactose disaccharide being converted into 2 glucose monosaccharides by the help of this enzyme. If galactose is an inhibitor, that means it's going to slow this reaction down, which means it's going to decrease the amount of glucose that's being produced. And the more significant the inhibitor is, the greater of an effect this will have. So if this something is a major inhibitor, that means it's really wrecking the effectiveness of this reaction, and we're going to see a lower glucose concentration. Now over here for your last 2 questions on the post lab, which, if any, of the compounds inhibited the activity of lactase? Anything that has a number that is considerably lower, that's something that inhibited it. And you know, you want it to be quite a bit lower, not kind of in the same ballpark, but you want it to be noticeably lower. And then down here, which carbohydrate from part 5 was the most potent inhibitor of lactase? What implication might this have for people who are lactose intolerant? People who are lactose intolerant lack that lactase enzyme. And so, just sort of use what you know about lactose and lactase and inhibitors, thinking about the nutritional diet of people who are lactose intolerant and how that pertains to these carbohydrates, galactose and maltose.